Meus queridos amigos, é um prazer hoje. Hoje a gente tem a presença da professora Aline Martin, ou Aline Martin, como seria seu nome verdadeiro. A Aline, ela fez seu treinamento básico na França, a Aline é francesa, depois mudou para os Estados Unidos e passou pela Universidade de Kansas, Universidade de Miami, e atualmente está na Northwestern University, em Chicago. Ela é pesquisadora básica, sempre envolvida com grupos importantes, né, no estudo do metabolismo mineral, os estudos seminais de regulação de FGF 23, a Aline já estava envolvida, e hoje ela é uma das coordenadoras do grupo que eu considero o grupo mais proeminente em termos de pesquisa básica translacional de DMO, DRC, no mundo mesmo, né? Então, hoje eles têm vários modelos experimentais, testando proteínas que são reguladoras da formação e da mineralização óssea, e também conseguem fazer vários estudos de colaboração com pesquisadores clínicos, fazendo justamente essa translação do lado experimental para o aspecto clínico. Então, aqui a gente vai aprender bastante. Eu sempre digo que os pesquisadores básicos são muito mais inteligentes que nós, médicos, porque eles conseguem enxergar muito mais profundo, né? Então, agora é a hora da gente aprender e, se possível, também ter perguntas ao final da apresentação. Aline, thank you very much for being with us today. So, please, you are ready to start? Are you ready to start?
Yes, sounds great. Thank you so much, Rosa. I mean, thank you for your kind introduction. I could make maybe a third of it, but and thank you so much for having me today. I will share my slides here so we can get started. And let's put this as a presenter mode. Okay, can you see my screen?
Yes, Ellen, it's okay.
And the pointer?
Yes.
Yes,
Yeah,
okay.
okay, perfect. All right, so I'm going to dive right in, and I thought that I would begin with an embarrassing question, which is, what is the best strategy to restore bone health in CKD? And as you're probably thinking about your own answer, I can tell you many colleagues would frequently say, well, we don't really know, it's complicated. Right. So, and that's kind of an embarrassing truth in some ways. So today, of course, I will not pretend that I have an answer to that, but I obviously look forward to your thoughts after the talk. So in the meantime, I hope to give you insights from studies that were performed in the lab, and that might help explore solutions maybe from a different perspective. So that's kind of my goal today. I have nothing to disclose. And so in the first part of the talk, I will start with reviewing pathogenic mechanisms of CKD-associated osteoporosis. So as many of you know, obviously, so this is CKD-MBD in a nutshell. And as kidney function declines, patients experience many secondary complications that heavily contribute to mortality. These include abnormal mineral metabolism, bone disease, vascular complication, cardiac complication. And today, however, I will only focus on the first two here, so altered bone and mineral metabolism. So this is our problem. CKD is associated with a significant risk of fracture. This risk is amplified with CKD progression and also with age, which is what you see here going through the colors. And so that's compared to the general population, which is the black line that you see here. So even post-kidney transplant, we continue to see a decline in bone mineral density in the peripheral skeleton, which you see here. And really, there's no therapy that specifically is designed to address impaired bone quality in CKD. And the main problem is that we don't fully understand the pathogenic mechanisms of bone disease in that context. So this recent JCI paper has given us some key mechanistic insights that might bring us closer to the answer. This came from a collaboration with Val David at Northwestern here and Rosa Moises, who you all know. And that came after years of RNA sequencing of bones coming from several mouse models of altered bone and mineral metabolism. And in these, we found that the transcription factor HNF4 alpha 2 was always heavily suppressed. And including in the mouse model of CKD, the call4A3 knockout, which you see here, for which we have down to a 99% reduction in its expression in bone. So Val reached out to Rosa, and what came out of this is let's sequence patients. And they did, and they selected patients with CKD with low bone remodeling or high bone remodeling, which you see here. And they showed that HNF4 alpha stood among the differentially regulated genes that contributed to impaired osteogenesis, impaired metabolism, and to cell death. And so consistent with this finding, going back to the mouse, if we repeat HNF4 alpha using a model of HNF4 alpha overexpression that we cross with the CKD mouse, then that corrected the bone disease, which you see here. And of course, this really does not mean that HNF4 alpha is the only factor responsible for bone disease, but it certainly plays a key role at the transcriptional level. So a few other studies, including these performed in pediatric CKD, indicated that osteoblast maturation is impaired in CKD, and this could be due to significant epigenetic modifications, which further points to an important osteoblast defect. So as a reminder here, bone, and you probably all know that, so bone is constantly remodeled during cycles of bone resorption handled by the osteoclasts, and bone formation and mineralization handled by the osteoblasts. And altered remodeling often results in bone disease. So for example, in postmenopausal osteoporosis, we're looking at too much resorption in the face of tudial formation. So in CKD, remodeling is also impaired, but it's actually more complicated because of this issue with osteoblasts. And as osteoblasts normally differentiate, some become osteocytes and support the mineralization process. But in human and mice with CKD, and these are studies that we did at different times, so I apologize if the scale is not the same for each picture, but osteocytes in CKD will appear more circular, and they have less cytoplasmic dendrites than the elongated healthy controls that you see here. And this was observed at all stages of CKD, so it doesn't really matter if you're an older person with early CKD or a young person with advanced CKD, the osteocyte morphology is the same. So we get the same observation. So if you isolate these cells and culture them in a dish for many weeks, so perhaps one of the most simple but exciting observation is that they retain mineralization defects in vitro. And that happens even if you give them everything they need to differentiate. So this study was done by collaborators on osteoblasts isolated from human bone biopsies, and they showed delayed and impaired mineralization after even after 4 weeks of culture. And we observed the same thing in my lab on osteoblasts that we isolated from our favorite CKD model, the call4A3 knockout, and all point to local persistent defects in differentiation and mineralization in osteoblasts. So osteocytes, they're the main cells that secrete a hormone called FGF23. And if they differentiate less, you would think that they would there would be less expression of FGF23 in these cultures. But on the contrary, we measured FGF23 mRNA expression, and we found that it was highly expressed in CKD osteoblasts. So we wondered if FGF23 could have direct effects on bone. And so there's been very few older studies actually hinting at possible direct effects of FGF23 on osteoblast. And most of them were done in non-disease contexts, and only one at the bottom right here was performed in a rat model of CKD in which they challenged it with dietary phosphate supplementation. And so in this model, it was a little bit complicated because the effects of FGF23 could not be dissociated really from the effects of PTH and other factors in vivo because of that phosphate challenge. So both FGF23 and PTH go up, and it's actually very hard in a model to disentangle. And so the reason it's hard, and I can make a little reminder of this, is because FGF23 works together with PTH and vitamin D to coordinate the regulation of calcium and phosphate metabolism. So the established function of FGF23 is to promote renal phosphate excretion and to inhibit vitamin D production in the kidney. But in CKD, FGF23 rises very early during progression and then exponentially as patients reach end-stage kidney disease. Once FGF23 rises, then as a result, vitamin D levels, which is the blue line, decline, and that then contributes to secondary hyperparathyroidism, which is the green line here. So for a time, all this contributes to maintain serum phosphate levels normal until the kidney is no longer able to sustain enough phosphate excretion. And finally, serum phosphate levels will increase. So the same thing is found in the COL4A3 knockout model I was mentioning earlier. And it shows a progressive decline in kidney function, which we measure with BUN levels. FGF23 starts to increase between 12 and 16 weeks. PTH increases at 16 weeks. And finally, we have serum phosphate that increases around 20 weeks of age. And so interestingly, the bone loss starts very early, around 12 weeks of age, and then it worsens from there. And this is accompanied by the reduced expression of pretty much almost every osteoblast differentiation marker in bone. So to give you a little bit of an example of or some context that relates to the study that was performed in a rat, if you challenge these mice with high phosphate diet, and we did this here for about 20 weeks, phosphate intake will accentuate FGF23 and PTH excess. So this is your high phosphate group in red here that has CKD2. And this is associated with aggravated bone loss that affects both the trabecular and the cortical bone compartment. And by RNA sequencing, we also showed that high phosphate diet will further modify the pathways that were already modified in bone in CKD mice on the control diet. So we actually did this study for a complete different reason, but I thought I would put it here because it really tells you how when you manipulate one parameter of mineral metabolism, all the others will kind of change to adapt. And this is kind of where you get stuck because you have a condition where you have high FGF23, high PTH, high phosphate, and who knows what else is modified, right? And as designed, it makes it very difficult to disentangle all their effects on bone. So in this next part of the talk, I'll spend some time talking about bone disorders during altered phosphate homeostasis. And to tackle the question of whether FGF23 would have direct effects on bone, we decided that we would take kidney disease out of the equation using non-CKD models of FGF23 excess. And these are models of hereditary hypophosphatemia. Contrary to CKD, FGF23 excess in these models is caused by rare monogenic mutations in the FGF23 gene itself. So in this case, it's going to be activating mutations, or in upstream regulators of FGF23 in bone, like FEX and DMP1. And these will be deactivating mutations. And in absence of kidney injury, FGF23-induced phosphate excretion results in severe hypophosphatemia, so low serum phosphate. So how it works is FGF23 binds and activates FGF receptor and clotho complexes in the proximal tubule, which inhibits then the expression and the internalization of sodium phosphate cotransporters that are in charge of phosphate reabsorption. As a result, we increase urinary phosphate excretion, serum phosphate decreases. So in patients, this results in severely impaired bone growth, impaired mineralization, which we call the somalacia, and rickets with the bone deformation that you see on these pictures. So these syndromes have been extensively studied in mouse models using FEX and DMP1 mutant mice that recapitulate what is observed in patients. Both models are phenotypically mostly identical. They show increased FGF23 levels in the circulation and hypophosphatemia. So the serum phosphate levels in a mouse is actually higher than what we see in humans. So this would be normal and this would be low. And it's generally accepted that the low serum phosphate is the main cause of osteomalacia in these models and of growth defect and rickets. But here I want to emphasize that while we clearly need phosphate for mineralization, more phosphate does not equal more mineralization. And I will show you a perfect illustration of this in this next study. So conventional therapy for XLH and ARHR consists in phosphate repletion in form of oral phosphate supplementation and calcitriol and vitamin D analog. So, however, it's been proven to be not very effective both in human and in mice because mostly FGF23 will increase further in response to oral phosphate, which results in higher urinary phosphate excretion. So whatever phosphate comes in will immediately come out. And this is a busy slide to kind of show you the same concept, but this time in wild-type and DMP1 knockout mice, where we performed a very detailed analysis after they were fed a control diet here, or 2% phosphate diet, so high phosphate diet for 6 weeks. And so similar to the CKD model I showed you earlier, the dietary phosphate further increased. So you have the DMP1 knockout in red on the control diet that shows already FGF23 excess. On the high phosphate diet, we further increase FGF23 levels. We further increase PTH levels. And together, these, and in absence of kidney injury, these keep doing their job and increase urinary phosphate excretion, which allows or leads to maintenance of serum phosphate levels at the low level. So basically, none of that extra phosphate that was in the diet went into the bone, and none of the bone parameters, or the bone growth, were rescued by the diet. And in fact, what we saw is a worsening of the cortical and trabecular bone mineral density. So coming back to studying the effects of FGF23 on bone, you can go both ways, right? So you can increase FGF23 like with the high phosphate, but it's proven to be not very optimal, or you can reduce FGF23. And this, in an animal model, can be easily done by a genetic deletion. And this was done by the group of Ken White in Indiana in the so the FEX mutant hype mouse model. And they used a COL 2.3 Cree, so COL1A1, that targeting specifically early stages of osteoblast differentiation. So that way they target a large pool of osteoblast in which they delete FGF23. And this led to the rescue of FGF23 levels, which are now just below normal, but still detectable. It rescued phosphate levels to normal. And as a result, the hype mice with the FGF23 deletion looked similar to the wild type controls in appearance, so which is what you see here. And they no longer showed rickets. So the shape of the bone was rescued. And there was this little bit of issue with trabecular bone. And there was no really explanation provided for this, but overall, they got a very nice rescue. So this is probably when you say, well, okay, but this could still be due to the rescue in phosphate levels. So you fast forward 7 years, actually, and in our lab, we repeated this approach in the other model of hypophosphatemic rickets, which is the DMP1 knockout. And instead, this time, to delete FGF23 in this model, we used the DMP1 CRE. So we targeted a smaller pool of cells, focusing on osteocytes mainly. And doing this, we didn't get a full rescue in FGF23 levels. It was about 80% rescued. And we had about a 50% rescue in PTH levels. And together, this was enough to reduce urinary phosphate excretion and to bring serum phosphate levels back to normal. So we're really in a situation where phosphate is normal, but we still have residual FGF23 and PTH excess. And so in that situation, despite the full correction of phosphate, okay, this I can skip. Despite the correction of phosphate, we only partially rescued the bone growth in the double mutant. You can see the shape of the bone here is not completely improved. The cortical bone porosity is only partially rescued. So was cortical thickness. The morphology of the growth plate was improved, but we did not improve bone mineral density. You can see a lot of residual osteoid in this histology section, and that suggests persistent osteomalacia. So correcting hypophosphatemia is really not sufficient to cure the bone. And at this point, we said, well, we have to look for phosphate-independent effect. So just to, as an apartheid here, I want to say it's not all about phosphate, except during the Gordon Research Conference, which I had the privilege to chair earlier this year, but now we have the chance to have Rosa Moises as the next chair. So I would highly recommend to convince her to take you with her at the next one. It's in 2027 in Texas, and it's a fantastic little gathering. So going back, in the next part of the talk, I want to discuss the direct effects of 23 on osteoblasts. And so if you recall, we have a model in which FGF23 and PTH are still a little bit elevated despite normal phosphate. And to separate the effects and to demonstrate that FGF23 has an impact on bone, what we did next is that we isolated bone marrow stromal cells from each of these mice. And then we cultured them for 3 weeks. And at the end of the 3 weeks, we performed bulk and single cell RNA sequencing. And in single cell RNA sequencing analysis, first, we were able to identify 4 very nice osteoblast clusters, ranging from osteoprogenitors to all the way to osteocytes. This is the pseudotime analysis that shows the same thing, osteoprogenitor in purple to osteocytes in yellow. And the next thing we do is segregate this by group. And we show that the DMP1 knockout cultures contain more osteoprogenitors and less osteocytes compared to the wild-type cultures, the controls, suggesting that the differentiation of these osteoprogenitors is altered in the DMP1 knockout. And interestingly, this is rescued when you delete FGF23 from these cells, so pointing to an effect of FGF23 on differentiation. So next, using this sequencing analysis, we identify genes that were modified in the DMP1 knockout, but no longer modified after FGF23 deletion. So using both the bulk sequencing here, we found about 4,000 genes that fitted that description. And in single cell, osteoprogenitors were the cells that showed the highest number of differentially regulated genes, about almost 800. And consistent with an effect of FGF23, the most represented signaling pathways in these genes were FGF receptor, ERG, PI3 kinase, and NFAT, which are downstream of the FGF receptor signaling, and osteogenesis and cell morphology. And we found this in the entire pool of cells by bulk and in the osteoprogenitor cluster, but not in these three other clusters, indicating at this point that FGF23 will target osteoprogenitors only. And so, to demonstrate FGF23 effects on osteoblast differentiation, we co-cultured cells from each genotype with their isogenic control, so wild type with wild type, a knockout with knockout, etc., or with cells that overexpressed FGF23 or that overexpressed DMP1. And so consistent with the RNA sequencing results, we now show reduced osteoblast differentiation in all the DMP1 knockout cells. This was partially rescued when we deleted FGF23, and consistent with the role of FGF23 on differentiation, all the co-cultures with FGF23 transgenic cells showed reduced differentiation, and that's regardless of DMP1 expression. So it works also in the wild type and the FGF23 knockouts. And finally, we decided that we would treat MC3T3 osteoblasts in vitro with exogenous recombinant FGF23 for a short time, so here over 2 days. And we showed a dose and a time-dependent acute reduction in the expression of osteoblast differentiation markers such as Osterix, RUNX2, and osteocalcin. And together, that's how we finally demonstrated that FGF23 directly inhibits the differentiation of osteoprogenitors independently of phosphatemia. So as a result, we kind of beat the odds because just a few months apart, we saw a bone on the cover of a non-specialty journal, and we also saw a solar eclipse. So we decided that once in a lifetime, bone can be the center of attention, and we felt very, very lucky. So in the time I have left, I wanted to discuss the relevance of this in CKD, which I think will interest you also. And so we went back to our favorite model of CKD, the call4A3 knockout, and we said, well, what if we tried the same thing and lower FGF23 just enough by deleting it from the bone and see what happens. And so for this, we used an osteoblast-specific CRE to delete FGF23 in osteoblasts. And what you can see is that that did not change impaired kidney function. BUN levels are still high. We got about a 50% rescue in serum FGF23 levels. That's a log scale here. So it was a pretty decent rescue. As a result of this, because you lower FGF23, then serum phosphate levels will go further up, which is expected. And all in all, PTH levels did not change. They were still high in the CKD model. So in the bone, depending on which parameter you look at, we reached a partial to a full correction of both the trabecular and the cortical bone loss. And that's by micro-CT analysis, and you can see this in those pictures as well. So this will hopefully be published soon, but in another approach that was already published a few years before that, we actually used DMP1 to lower FGF23. And so I talked about DMP1 in the context of the DMP1 knockout, but I didn't explain exactly how it works. So that would be DMP1 in pink. We think so. DMP1 is an established inhibitor of FGF23 that's produced in osteocytes, right? We think that's how it works. DMP1 binds to FEX at the osteocyte surface or osteoblast surface, and it represses the paracrine activation of FGF receptor. And so when DMP1 and or FEX are mutated or deleted, then FGF23 will go up. So in mice and in human with CKD, FEX is expressed, is there, but in mice, DMP1 expression in the bone is reduced throughout CKD progression, and this is what you see here on this graph. So we decided to repeat DMP1 by crossing these mice with a DMP1 transgenic mouse. And so that would be the pink group here, with now very high expression of DMP1 in the bone, specifically in the bone, and with CKD. And we have a little bit of expression in kidney and heart, but nothing compared to bone. So similar to the study where we deleted FGF23 in the osteoblast, here with DMP1 overexpression, we kind of reach exactly the same pattern. So we lowered FGF23 levels by about 50% here and here. As a result, serum phosphate will go further up. This did not impact PTH or kidney function. So PTH levels remained elevated, kidney function remained as bad. And so by lowering FGF23 and increasing DMP1, we observed a nearly full correction of the bone loss. So the cortical and the trabecular bone BMD, bone mineral density, were fully rescued in mice with advanced CKD. So in addition, and if you recall these osteocytes that I showed you at the beginning of the talk, we also observed a full correction of the cortical bone osteocyte morphology. So from round shape, they go back to elongated shape, more cytoplasmic dendrites. And this was associated with reduced apoptosis compared to CKD osteocytes, which possibly suggested improved osteoblast differentiation. So indeed, when we when we brought those cells in vitro, we also rescued mineralization. So you see impaired mineralization in the CKD group, and that's completely rescued here, which points to a local effect on osteoblasts once again. I apologize for the interruption here. So, and finally, as a little bonus, despite hyperphosphatemia and hyperparathyroidism and CKD, and in addition to improving bone, reducing FGF23 prolonged the life of mice with CKD by 2 to 4 weeks here and here, compared to the control mice with CKD in both cohorts. So either if we delete FGF23 or if we overexpress DMP1. So now it was shown, as you know, that the mortality risk in CKD increases with ascending FGF23 quartiles, possibly due to FGF23 inducing cardiac hypertrophy. And we have additional data that I won't have the time to show today, but some is already published, and they show the improvement of the heart phenotype in response to lowering FGF23 levels in these cohorts. So in terms of therapeutic outlook for CKD, what does it really mean? In recent years, there was a new therapy that was developed and approved for the treatment of XLH. And so it's called burosumab. It's an anti-FGF23 antibody that blocks FGF23 function. And for patients with XLH, the burosumab that's shown in blue showed superior efficacy, improving rickets severity score and maintaining serum phosphate levels in the normal range, which you see here, compared to the conventional therapy. So at this time, we don't really know, however, that what happens with bone remodeling and what happens with FGF23 production in bone. So I guess that's pending longer trial. I'm looking forward to see that. And in CKD, blocking FGF23 function has created some controversy in the past. And if you recall, there was this one study published in JCI in 2012 that used burosumab in high doses in rats with CKD, so 5-6 nephrectomy model. Unexpectedly, because of this high dose, mortality was very, very high due to aggravated hyperphosphatemia and high toxicity. But I hope that with the data I showed you today, I convinced some of you at least that perhaps it's actually time to rethink this idea. And if we, for example, targeted a smaller CKD population where that would not be at risk of hyperphosphatemia, like patients on dialysis, maybe that would be an interesting thing to do, or using a less aggressive approach in which we don't abrogate FGF23 function completely, but maybe lower levels, just enough to get the bone and heart benefits that we saw in the mice. So coming back to our embarrassing question, perhaps it's time to pick a dare instead, and it might be less embarrassing than the truth in the end. So on that note, some thoughts to conclude, and I think we need to recognize that altered osteoblast differentiation contributes to CKD-associated osteoporosis. So we're not just talking about resorption anymore. Formation and mineralization, I think, are at the very root of the problem. There's evidence that supports direct inhibitory effects of FGF23 on the differentiation of osteoprogenitors, and I think it's a nice new avenue for diseases associated with FGF23 excess. And finally, lowering FGF23 in CKD, at least in mice, prevents bone loss and other important adverse outcomes. I think it's also important to note that this happens despite persistent hyperparathyroidism and hyperphosphatemia. And so with this, so this is our team at Northwestern. You might recognize this guy in the back. As you can see, we feed him well. He's very happy and he says hi. And so I will thank my collaborators and our sponsors. And I thank you all for your attention. And I will take questions.
I will just open the chat. And please, everyone, por favor, todos que coloquem as suas perguntas, sintam-se à vontade de fazer as perguntas, porque essa é uma oportunidade, creio que única, né, de vocês poderem conversar com alguém que está envolvida na pesquisa básica, que daqui a alguns anos vai provavelmente ser a translação para o que a gente vai estar fazendo na clínica. Aline, I would like to start asking if we take your hypothesis, and I think that it's interesting, but I'm a little afraid that because of the data that we have in the past from Chalhoub study. Do you think that a combination of burosumab and a very low phosphate diet would be the perfect direction to go forward?
Yeah, that's a great point. I think, I'm not saying that FGF23 is the only thing we should, address. I think we're starting to have data that point to, other roles of FGF23, and if I think a combination would be perfect, because then maybe you can, reduce FGF23 or block a little bit of FGF23. You don't want to reduce it completely, because otherwise, you take the risk of having phosphate and so high. So you don't want that. But if we find a way to just find, this little window where it's at the right level, and then maybe combine that, with something else to address hyperphosphatemia, hyperparathyroidism, then we might be in a nice little condition where you prevent maybe the bone disease, you prevent maybe the cardiovascular outcomes. So I think you're right. Yeah, it's something to think about. But we need more clinical studies for that. That's the thing. Like, I'm showing animal models, obviously, but I think we need more clinical studies to explore that aspect.
Lynn, you think that the MP1 could be a possible treatment to this situation? Or?
The short answer is yes. The long answer is we've actually taken steps to try to develop DMP1 as a treatment. It's very early, but that's a great point. Yes, I think with DMP1, the advantage also of DMP1 is that we know its established function to also improve mineralization independently of lowering FGF23, right? So you get the direct effect on bone, and then you have the benefit of lowering FGF23 in the process. And so indirectly, then you can actually reach, to other problems like the heart, indirectly through FGF23. So we'll see where that goes, but yeah, we're actively involved in the lab in looking at that.
There is a question here from Ediva Nilsson from Recife, Pernambuco, and he says that we know that clotho deficiency is a hallmark of increased kidney function. Is there any evidence that modulating clotho could lead to a change in bone phenotype?
Yeah, that's a great question. So we haven't directly tested. So in CKD, we know that clotho expression goes down, right? So there's some clotho deficiency. In the bone, there has been studies that show that clotho is actually expressed. In our studies, we haven't tested if clotho is involved in mediating the effect of FGF23 on osteoblast directly. So that's something that we still need to look into. The fact that we see FGF receptor, ERK, PI3 kinase, and NFAT signaling modified in the rickets model kind of suggests that it might be clotho dependent. Maybe modulating clotho locally could be an approach. I don't know. Like that would still have to be done. Yeah.
Aloysio, I see you. Would you like to make any
Hi. Hi, Aline.
question?
Good to see you again.
It's good to see you too.
We met in Houston. Yes, there are some concerns about burosumab adverse effects in cardiovascular disease. Would you translate your hypothesis to vascular smooth muscle cells, your hypothesis on osteocytes, and all this orchestra of DMP1 and FGF23 phosphorus and so on?
Yeah, I think so. That's also an angle to consider, of course. I think so one thing that we haven't looked at, because the model does not allow us to do it really, is look at vascular calcification, right? So obviously you see that by lowering FGF23, whichever way you go, right, serum phosphate levels go up. That's potentially a concern for aggravating maybe the development of vascular calcification. That's totally agreed. So that would be something to look at. And I think that kind of emphasized the point that Rosa was making earlier, is that maybe that would be important to, if we address FGF23 levels, maybe to address it in combination with something that would kind of lower phosphate levels too, to prevent ultimately, also the development of vascular calcification. So in the mouse, so these models, you can push phosphate very high, and unless you put them on a very high phosphate diet for a long time, then they don't develop vascular calcification. It's very hard to do. They don't calcify. They don't want to calcify. No. So I think it also needs to be investigated a little bit further. The thing is, in the CKD model, we get such a beautiful rescue and prolongation of life also, right? I mean, I think that's also important to emphasize, is that despite phosphate going higher and PTH going so high, then the mice still live longer. So what if we manage phosphate a little bit better? Maybe they would live even longer, right? So yeah, so I think that's a great point.
Okay, thank you.
More questions? I have other one.
Go ahead, Rosa.
Yeah, just considering usually our rats and mice, they are living organisms that are continually growing, right? And this is different from us, the humans. And in the CKD, the human CKD model, bone sclerostin expression is something that occurs very early. I don't know if it goes at the same point in the murine model. I don't think so. As far as I remember, I saw higher sclerostin expression when we were able to decrease parathyroid hormone through parathyroidectomy, unless sclerostin is not there.
Yeah.
So considering that sclerostin there, and probably looking from your data and Renata's data in humans, you have this decrease in the MP1 action or expression. Could you bring us some hypotheses that could connect sclerostin to the MP1 and FGF23? I can't. This is why I'm just...
Yeah, I don't have a good, that's a good point. I don't have a good answer for you there. I think so far, in all the screens that we've done by RNA sequencing, in all those different models in bone, there was not really anything pointing strongly to modified sclerostin. So we didn't really, look into it too much. I'm not sure. Yeah, I'm not sure that it's actually involved in any of these models, at least. But then in human, maybe. Yeah, I really don't have a good answer for you.
Oh, me neither.
In the mice, yeah, in the mice, in the mice, at least, we couldn't find any data that would point to a strong role of sclerostin. So,
Aline, I try to explain you my question. You said that lowering FGF 23, you can prevent bone loss despite persistent hyperparathyroidism and hyperphosphatemia. Did you see this same observation in heart tissue? It is the same for heart because we know that hyperphosphatemia is enough to produce a hypertrophia, a myocardial hypertrophia. It is independent of FGF 23 or what is the most important, FGF 23 or hyperphosphatemia in this moment that you try to separate these both?
Mm-hmm. Yeah. Yeah, that's a great question. So, and a very interesting one too. I think, so in these models, in the exact same mice, right? So here I showed you the data on bone, but we've also analyzed the heart by echocardiography. We've recorded blood pressure, and in the DMP1 cohort, this was published. So if you want to have access, to this data, you can. That's it's been like several years already, but what we found is that the mice with CKD develop cardiac hypertrophy. They develop hypertension. But when you lower FGF23 with DMP1, then you rescue the hypertrophy. However, the hypertension stays there. And as you saw, serum phosphate levels are higher, serum PTH levels are higher, or still high, I should say. So I think at least for a time, because these might still die, right? And we don't really know what they die of. Is it kidney failure? Is it heart failure? So I think at least for a time, we prevent the development of cardiac hypertrophy by manipulating FGF23. And even though we worsen phosphate and the rest, still is the same. And we don't improve, kidney function. We don't improve any of that. So I kind of tend towards, FGF23 is more important. Also, there's like a strong literature showing, the direct effects of FGF23 on the heart. And yeah, I think FGF23 is a very strong player into the development of hypertrophy. Is it the only one? Probably not, but in this cohort at least, phosphate did not really. I don't want to say did not matter. I think it matters, right? Because, like I said, maybe if we had lowered phosphates, then we would have an even better phenotype. But just by modulating FGF23, that was already a very strong effect.
Okay, thank you.
Rosa, we have another question in the chat.
Yeah, it's from Julia. Julia is one of she's Vanda's student in Australia. Actually, she's very interested in mineralization defects. She is the expert in the group in mineralization defects. She says, "Hi, Aline, thank you for your talk. Based on your findings, do you think that selectively lowering FGF23 in bone could be a feasible therapeutic strategy in humans with CKD and BD, or would the risks of hyperphosphatemia still outweigh the benefits?" I think you comment on that, and in that context, do you think that BMP1 restoration would be as a potentially safer or more upstream approach that than ablating FGF23 itself. So I think that it would be safer. Yeah.
Yeah, I think you're making a great point. And that's also a great question. Thank you so much. And I totally believe that with the MP1, you get several advantages, I think. Because first, I believe you would get a more bone-specific effect. Because DMP1 will will go into the bone matrix. And we've shown this in mice, even if you inject it. So that's in that previous paper that we've published in Bone Research. If you inject DMP1, then you can detect it. It penetrates the bone, so it goes to the bone. And also, I think, DMP1 is going to have effects on not just FGF23. It's going to have effects, like I was saying, on the mineralization itself. So I think you get a little bit of a benefit there where maybe you lower a little bit FGF23, not completely. You don't block it completely, its function. You still have, enough FGF23 maybe to get rid of the phosphate that you don't want at the kidney level. And then you improve the bone. So I strongly believe, yes, DMP1 has a lot of potential. We need more studies. We're working on it. So hopefully soon I'll have more data to show you.
Thank you.
So do you have any data besides Renata's data on DMP1 in humans? I can't remember. So I think she was the only one who has shown DMP1 defects and decrease in human samples, right?
Yes, and I think so you had a paper, I think, where you showed patients with lower DMP1 are more susceptible to fracture, right? Is that the
Yes.
paper you're referring to? Yeah,
Yes.
yeah.
It's Vanda's paper. I am in the group, but yeah,
Vanda's paper. Sorry.
Yes.
Yes. So yeah, I think I believe that's the only one. I don't think anything else has been shown. Kate Westling actually had some immunostaining of bone biopsy. So they showed some DMP1 expression, and so the interesting part is that in pediatric CKD, it doesn't seem to be quite the same as in adult CKD in human, because DMP1 expression in kids seems higher in CKD than compared to healthy controls, as opposed to adults where it seems lower. So there's probably something interesting there. I think maybe in kids, since you have all these bone development still going, maybe they need, a lot of DMP1. I don't know. So, but there's not much out there. Yeah, that's true.
Okay, and there is a final question, I think, because we take so much advantage of Aline, but I guess it's from Emilia, right? Emilia Soeiro, also from Pernambuco, and she is thinking about possible therapeutic targets and taking into account the ratio and the interaction between FGF23 and the MP1 expression in osteocytes probably change across the different stages of CKD. So do you think that there are specific implications for bone mineralization in each stage? It's a very good question. Yeah.
Wow, that's an excellent question. Yeah. So if we look at CKD, so I have obviously our CKD model in mind, right? And we looked at different time points. So FGF23 increases very early, like it increases before everything else changes in CKD. And we see actually a bone phenotype very early on too. So I don't know about, so DMP in our case, in this model, we see lower DMP1 across the board. So it's interesting because even early on, so we see a reduction in DMP1 even before we see any changes in FGF23 and bone, which I think to me is kind of head scratching, because you would think that maybe lower DMP1 early on would aggravate this FGF23 excess very early on too, but it doesn't necessarily is the case. So I think there's really, however, this very nice correlation between the rise in FGF23 and the decline in bone parameters. So I think we can probably maybe take DMP1 out of the picture. And I like to think that maybe, early on the bone defects might be caused, at least in part, by the rise in FGF23. I don't know if that answers your question, but yeah, I think it's hard to have an answer for these things. Yeah.
Thank you. Probably looking in the human samples and early CKD, and this can bring us some ideas. Yeah.
And actually, I mean, we actually have tried another model, which I didn't show today, but I think it will also get published soon. So we did an AKI to CKD transition model where surgically you induce kidney injury. And then we looked at many weeks after the surgery procedure. So it's a bilateral ischemia reperfusion injury model. We looked at the bone. And it's interesting because, so we looked at different time points, and it's interesting because you see that the kidney injury and the kidney function progression stays a little bit stable, but then you start to see these modifications in FGF23 and bone, but not PTH. And PTH occurs very late. So we also have like this little window where you see the bone phenotype evolving in phase of changes in FGF23, which is also nice. So we'll see what mechanistically, we don't have that data yet, but we don't know what's going on. But I think it could be also an interesting model to look at.
Okay, I think we have more than 1 hour, and it was fantastic. So I will call Melanie and Leandro to close the session. And thank you so much, Aline. It was a pleasure to have you with us today. Almost 50 attendees, which is a
Pleasure
record
was
for
all
us.
mine. That was so great to see all of you and hear your thoughts. I think that's great. That's fantastic. Thank you so much.
Melanie, you are muted.
Yes.
Aline, thank you very much for your great talk. And I want that you come again and stay with us. And now we know about your work, and you can prepare many questions to you. Yes, in the next time. Okay. Thank you.
Absolutely.
It's a pleasure.
Thank you so much, Melanie.
Lane, thank you by your time and your divide your knowledge with us. Thanks a lot once again. Thank you. Thank you. Bye bye.
Thank you, Leandro.
Thank
All
you.
right.
Thank you. Thank you.
Thank you, Rosa. Thank you, everyone.
Great holiday. Bye.
Thank you.
Bye.
Bye-bye.
Bye bye.
Good night for all.